Hello, I'm Alysson Muotri, Director of the UC San Diego Stem Cell Program, and this is the Sanford Stem Cell Clinical Center. So why do I have these little guys here with me? One of the future hopes for treating leukemia is using the cells that give rise to blood, hematopoietic stem cells. And these guys have helped us to understand a lot about how the cells do that. UC San Diego's David Traver uses these zebrafish and other model organisms to understand exactly how these very special blood stem cells are born, with the goal of duplicating that process to cure diseases. David shared why and how the zebrafish has helped us, and what they have revealed that is leading us toward cures.
We study the stem cells that make blood, which are these very rare stem cells in our bone marrow. Despite their rareness, they have really impressive proliferative power. They can generate all of the hematopoietic lineages we have for our entire life. So the thought in the field is that leukemia, and probably all cancers in general, occur due to mutations within these stem cells. Any one of those lineages can be transformed to give rise to leukemia. You can have T-cell leukemia, B-cell leukemia, there are granulocytic leukemias, myeloid leukemias, lymphoid leukemias. There's as many types of leukemias as there are lineages. So the gold standard treatment for leukemia is bone marrow transplantation, really the first stem cell treatments in the clinic. This has been around since the late 1950s. When it works great. If you have an identical twin, for example, then bone marrow transplantation can work exceptionally well. But most people don't have that, right? So in that case, you can get all sorts of immune rejection. So that's why the field has been looking for a long time to try to make patient-specific hematopoietic stem cells. It was sort of a pipe dream for a long time, but in the last 10 years, it really has become reality because of Shinya Yamanaka's findings that you can take essentially a handful of factors, put those into basically any adult cell type, and transdifferentiate that cell to something that is pluripotent. And that cell that you can essentially just make from a swab from the cells in your cheek, they'll become the so-called induced pluripotent cells, or IPS cells, and those cells then have the potential to make any cell type in your body. So that part is actually very straightforward. We could make IPS cells from you or me in a matter of weeks. That part is very trivial now. The challenge for the field is then to recapitulate development. So you can't transplant iPS cells by themselves because they're too naive, they're too immature. They don't know who they're supposed to be. So what you need to do is then take these cells in vitro and instruct them to become certain types of cells. There are liver stem cells, hematopoietic stem cells, neural stem cells. And those are the cells you'd want to transplant into somebody, right? Because to keep a patient alive for years, you have to have stem cells being transplanted. And so people have been trying to take these pluripotent cells and instruct them to make hematopoietic stem cells for 30 or 40 years now, and we just haven't been able to do it yet. I think the main reason for that is because we just don't understand how the embryo does it, right? We don't know the full recipe. And so the major goal of my group is to try to use lessons from the embryo, mostly the zebrafish embryo, because it presents these really nice opportunities to do imaging. Another reason that developmental biologists love the zebrafish is because they develop extremely quickly. The hematopoietic stem cells, for example, in humans take months to develop. During zebrafish development, we think these initial specification events are happening about 12 to 14 hours after fertilization. You can actually watch these things happen sort of in vivo, in real time, using confocal imaging and time-lapse imaging. We actually had the first demonstration of how exactly hematopoietic stem cells are born because we simply just imaged these embryos. We can actually watch the process of hematopoietic stem cell formation. And we saw that these hematopoietic stem cells actually derive from the floor of the dorsal aorta. The dorsal aorta is the biggest blood vessel during development. And interestingly, there's just a population of these endothelial cells that line the dorsal aorta that then transform in this very interesting process to hematopoietic stem cells. And so what you can see in this movie is that you see these endothelial cells that are forming the floor of the dorsal aorta. They'll change their shape. You'll see the cells become a round cell. Over the next few hours, you can see that cell then lose its contact with the endothelium and actually enter circulation, going in through the roof of the vein. Those hematopoietic stem cells will then go to the specialized structure in the tail, where they'll start to differentiate and make adult blood cell types. This is true not only in a zebrafish, but it's true in a mouse, it's true in people. So no matter what kind of vertebrate animal you are, your hematopoietic stem cells come from the endothelial lining of your dorsal aorta. And so once we saw that, we then knew, okay, this is the event we need to study. This is the event we need to understand, because this is the first time we've actually been able to see the birth of a stem cell. And so what we've been trying to do since then is essentially roll the movie even further backwards to understand where those cells come from, right? And when do those cells know they're going to be that cell? So one of our goals in the lab is to know what factors are needed to make a stem cell, what order those factors are produced in, the relationships between those genetic hierarchies. We've made some surprising discoveries. During development, these cells make direct contact with the bottom of a structure called the somite. Somites are these big chunks of tissue that are along the entire developing embryo. They later give rise to skeletal muscle, all of your vertebrae. The cells that are fated to go on to become the hematopoietic stem cells, they crawl and make direct contact with that sort of bottom face of the somite. If we can understand what the somite is doing, we can essentially just short-circuit the process, take the factors we know that are key and add them to the cultures at the right time, to try to push those cells to become the right lineage, basically. In this way, we've also discovered some other surprising things. In the adult, if you get a wound, you make things like tumor necrosis factor called TNF. In the embryo, we found just sort of serendipitously that you also need this pro-inflammatory factor to help generate hematopoietic stem cells. So we just have to understand what factors are needed, the order in which they're needed. Then we can try to, essentially distill those factors to add to these culture systems to try to push those cells to the right place. And so the 10-year goal, basically, is to be able to make patient-specific stem cells. If you have leukemia, we want to be able to irradiate you or treat you to wipe out the leukemia, also wipes out your normal blood-forming system. So the idea is we would take the cell from your cheek, transduce it with these factors. In a period of a couple of weeks, we can make your iPS cells. We can then take those iPS cells and generate your own hematopoietic stem cells that can then be put back into you. Make all again of those hematopoietic lineages you need and stick around for the rest of your life. So that's the goal. Patient-specific stem cell transplants.